Hello students, this is Dr. Sujata Magdhum, Assistant Professor, Department of Zoology, KTHM College, Nashik. I'll be dealing with immune responses, that is humoral type of immunity, and this is once again from the first chapter, that is Introduction to Immune System. Last lecture, we tried to understand the definition of immune system and broadly discussed about two different types of immune responses, which are mainly defined as cell-mediated immunity and antibody synthesizing immunity, which is called as humoral type of immunity. Further, we discussed in detail the cell-mediated immunity. Today's lecture, I'll be going to deal with only humoral type of immunity. To begin with this session, you need to understand what is humoral type of immunity. Humoral type of immunity is basically concerned with formation of antibodies, and this formation of antibodies is seen occurring only when the antigen enters into the body and it triggers a response with immunocompetent cells. This is achieved by interacting the antigen with the immunocompetent cells. Now this interaction leads to processing of antigen, and this processed antigen further helps in stimulating the activated immunocompetent cells to secrete the antibody. This is in general the short sequence of events which are observed in development of antibodies. There are four different levels at which one can understand or one can say the synthesis of antibody occurs. That is, the first one is called as the lag phase, second is called as the log phase, third is called as the plateau phase, and fourth one is called as the decline phase. Now let us see each of them in detail. Now consider a specific antigen making its entry into the body. At that moment, because there is absence of specific type of antibodies synthesized by this antigen, we can say the titer or the level of antibodies into the body will be totally negligible or absent or zero, one can say. Hence, graphically if I plot, we get a straight line, and this is called as the lag phase. Now in this lag phase, it is a long time period required wherein antigen makes its entry into the body, then it gets well distributed, and then there is an interaction of this antigen with the immunocompetent cell. So this time consuming process will be causing delay in synthesis of antibody, as a result of which the level of antibody, if I measure in this phase, it will be less or it will be almost equivalent to zero. Once the interaction is established between the antigen and that of immunocompetent cell, the immunocompetent cells will get stimulated, and as a result of which there will be secretion of antibodies seen. Now this secretion will be seen in a increasing manner, or you can say it enters into the log phase. So at log phase, if I measure the antibody, then I can say that there is huge increase in the volume of antibody against this specific type of antigen. Further, the number of antigens which has made their entry and interacting with the immunocompetent cell, as it remains constant in this third phase, which is called as the plateau phase, I can say the amount of antibody synthesis, it remains constant. This is because both the antigen as well as the activated immunocompetent cells, they are at equilibrium. Further, the antibody which is already synthesized, it will show its interaction on the antigen itself, as a result of which there will be elimination of antigen. Obviously, the number of antigen are getting reduced, and at the same time, the amount of antibody is also going to get reduced, because there are two reasons. First of all, the antibody which is synthesized, it is acting on the antigen, and because the number of antigens are getting reduced, there will be less chance for this antigen to interact with the number of immunocompetent cells. Hence, the amount of antibody which are synthesized in this fourth phase, that will drop down, and this is called as the decline phase. This can once again come to the level 0. Okay. So this is how the antibodies, they are synthesized and they are maintained into the circulation, right? Now we'll see primary and secondary responses. Now how does primary and secondary responses we get into the body? It is only when the antigenic stimulation is observed. There are two main phases or two main responses which are observed commonly upon the entry of antigen, and that is called as the primary response, and the second one is called as secondary response. In case of primary response, we can say that this is slow, sluggish, very short-lived, and it has a very long lag phase. It is shown graphically here. If you see the remaining, you can make out the difference that the lag phase for primary response happens to be very long as compared to any of the responses which are shown by the same antigen, right? Then, secondly, we can say when this primary response is getting noted, antibody formed is always IgM type. That is, immunoglobulin M type of antibodies are generated, right? The second type of response which we get to know is called as the secondary response. Now, this secondary response is very prompt. Till here, it is the primary response what we get, which has a long lag phase. Then there is a log phase. Then there is a plateau, and once again there is a decline. After decline, if there is second chance of infection of the same antigen, then immediately there is shoot up in the synthesis of antibodies seen. The reason here is because there is already sensitized cells which are immunocompetent memory cells, and these memory cells will come into action, leading to synthesis of antibodies. As a result, the titer will immediately rise. So this second response of antigenic stimulation will lead to secondary responses, which is very prompt, powerful, and long lasting. And here I can say that the lag phase is almost negligible, or you can say very short, and there is higher level or higher titer of antibodies, because already the memory cells which were triggered with the previous experience of the antigen will start secreting the antibodies, and the new antibodies will also be synthesized because of the second infection by the same type of antigen. Now, when this is the situation, we can say the amount of antibodies which are generated here, they are of IgG type, right? After completion of this IgG type, we will now go into the next part of our discussion, that is called as the negative phase. Now, to begin with, we can just summarize the two events what I have discussed, that antibody responses. Now, these antibody responses to initial antigenic stimulation differ both qualitatively as well as quantitatively with the same antigen. This response can be categorized as the primary response and the secondary response. I have already told you the primary response is slow, secondary response is prompt, primary response is sluggish or short-lived with long lag phase, secondary response is powerful, prolonged with short lag phase. In case of primary response, there is less amount of antibody synthesis occurred, whereas in case of secondary response, the quantity of antibody synthesis is more. This is only because of pre-existing memory cells, right? Duration of log phase and persistence of antibody varies with nature of antigen. Example, in case of diphtheria, the lag phase in primary response longs for 2 to 3 weeks, whereas with pneumococcal polysaccharides, it can be seen within few hours. Now I would like to tell you all a very interesting fact about the vaccines. The first injection which is always given is called as a priming dose, and this helps in sensitizing the immunocompetent cells which are responsible for production of antibody. Effective levels of antibodies are seen only after subsequent injections of antibodies. This is the only reason why non-living vaccines are always given in multiple doses, and these are defined as the booster doses. With the live vaccine, single dose is sufficient as multiplication of organism occurs into the body, and that produces continuous antigenic stimulation, and that leads to the priming as well as booster doses. Whereas when an antigen is injected into an animal already who is carrying the specific antibody, there is a temporary fall into the special type of antibodies which are synthesized. Now this is because the antigen binds with antibody, and this phase is called as the negative phase, right? Then the negative phase is followed by increase in antibody titer. It is suggested that this could be a prophylactic vaccine which is administered so that the host is exposed to the infection. Therefore, theoretically, there are chances of lowering of specific type of immunity. But in actual practice, this is little doubtful. Okay. Then, now we are going to understand the fate of antigen. To begin with, understanding of fate of antigen, you need to understand what does it mean. It means that here I'll be discussing what is the effect of on this antigen when it enters into the body. So let us begin with our discussion. Fate of antigen totally depends on its physicochemical nature of the antigen, its doses and route of entry. It also depends on antigenic stimulation, which means that whether it is stimulus, or that means it is primary or it is secondary. The first one, that is dose or load. You all know that the specific load of antigen is very essential for development of disease. Supposingly, the less number of antigens are seen entering into the body will not be causing any kind of disease, because immunocompetent cells, they will kill the antigens, and therefore the effect of antigen will not be observed. Whereas an appropriate quantity of antigen when it enters, probably if the immunocompetent cells are weak, obviously the antibodies synthesis will be lowered, and as a result of which there will be development of disease. Hence we can say that the viral dose, or you can say the viral load or the bacterial load, is very much essential in showing its influence in development of disease. Further, the type of antigen, there are two basic types of antigens, that is particulate type of antigen and soluble type of antigen. Now these two different types of antigen are very essential in showing its action. In the sense, the particulate antigen will be removed from the body or from the circulation, and this removal, it occurs in two different phases. The first phase is called as the phagocytic phase, and the second one is with the help of synthesis of specific antibody. You know that any particulate antigen which is entering into the body will be engulfed by the phagocytic cells like the natural killer cells, macrophages, dendritic cells, or the other monocytes, and this is showing a non-immune response wherein the engulfed pathogen by the action of these non-specific type of immunocompetent cell will help in killing. So we can say because the antigen is been engulfed by these immunocompetent cell, the action is phagocytic action. Sometimes the particulate antigens are removed by the action of specific antibodies, and here specific antigen antibody complexes are formed, and then such complexes are removed by the process of phagocytosis, right? Sometimes it is also seen that the second type of antigen, that is soluble type of antigens, are seen into the circulation. Now this soluble type of antigens which are in circulation, they are eliminated by three different phases which are namely defined as equilibration phase, metabolic phase, and the phase of elimination. The phase of equilibration consists of diffusion of antigen to extravascular spaces. During metabolic phase, the level of antigen falls due to catabolic activities, and during immune elimination, antigen-antibody complexes are formed. Such complexes can cause even the tissue damage and are responsible for development of immune complex diseases like serum sickness. In case of serum sickness, certain specific proteins when they are administered, then similar to allergic responses, the rashes will be seen developed onto the body of the recipient, and so this leads to development of mild fever and occurs only on administration of proteinaceous medicines. This is a sort of allergic response, we can say, right? Then the speed of elimination. Speed of elimination of an antigen is related to the speed with which it is metabolized. For example, protein antigens are eliminated within a day or a week, whereas polysaccharide antigens, that means the antigens which are made up of polysaccharides, they get eliminated in months or probably it can take years. The best example is an example of pneumococcal polysaccharide which persist for nearly 20 years in the body of human, right? So this defines the speed of elimination in case of host body. Persistence of antigen to immunocompetent cells. Now you know that this occurs in two different ways. There are two different types of cells which are responsible for elimination of antigen, and those cells are macrophages and dendritic cells, right? Macrophages and dendritic cells, both of these, they are seen present in the lymph node follicles. Antigen processing by macrophages helps in bringing about primary immune response, where the antigen captured by the dendritic cells of lymph node, it occurs in presence of pre-existing antibodies. Both macrophages as well as antibodies, both macrophages as well as the dendritic cells, they present antigen in native or processed form at the cell surfaces or in the body at the areas where there is large quantity of lymphocytes present, and this is essential to ensure appropriate lymphocyte to recognize the antigen and to show the interaction so that the immune response will be observed, right? Further, the route of entry also plays a very important role. Say for example, as antigen are introduced intravenously, then they are localized in the kidney, liver, spleen, bone marrow or lungs. right? These are broken down by reticuloendothelial cells and are excreted in the urine. Nearly 70 to 80% of such intravenous antigens, they are eliminated in a day or two. In contrast to this, whenever there is subcutaneous entry of pathogen or antigen, then these are mainly localized into the lymph node and very small quantity is seen observed into the spleen, right? Now let us move into the actual process of antibody production. We'll brush up the basics of this. You know that antibody production is basically seen only when B cells they are triggered or they are stimulated. Now these B cells by themselves are unable to produce antibody unless and until these B cells they have the surface receptors which are of IgM and IgG type, right? So such immunocompetent cells will have IgM and IgG type only when they become mature. So in case of immature condition, the antigen will interact with B cells which are immature and they will transform this B cell into mature B cell. After transformation or maturation, these mature B cell they get converted into the memory cell and the plasma cell. This phase of development of memory cell and plasma cell is related to clonal proliferation as well as blast transformation. Memory cells they enter into the circulation, whereas the plasma cells they are responsible for synthesis of antibodies. Now, the antigens which are responsible to bring about this kind of sequence in synthesis of antibodies, they can be categorized into two types. That is, the antigens which are thymus dependent or thymus independent antigen. Thymus dependent antigen, they require processing of antigen which is taken care by T cells. So, such processed T cells processed antigen by T cells will be donated to the B cells and that will trigger the response in synthesis of antibody. In the same way, there are certain antigens which does not require any processing by the T cell, and the distinction between these two makes us understand the process of synthesis in detail. The examples of thymus-dependent antigens are erythrocytes, serum proteins, and protein hapten conjugates, whereas polymerized flagellants, ferritins, or polysaccharides, they don't require the support of T cells for triggering the immune response into the B cells, and directly the synthesis of antibodies can be observed. Now this slide, it shows you the actual process of antibody production. Focus on to the attention where I am moving the cursor and then we'll move into the pictorial representation. Antigens which are entered into the body with the help of APC, that is antigen presenting cells. Example of antigen presenting cells are many macrophages or the dendritic cells. These they get activated, right? This activation means the antigen has entered into macrophages and dendritic cells, and into the cytoplasm of macrophages and dendritic cells, this antigenic processes, metabolic processes will lead in development of certain surface proteins which will be expressed onto the surface of APC, and that will be seen always in association with MSc II molecule, right? Here you can see that there is a red spot shown which is a processed antigen. Antigen has entered into the macrophage, into the cytoplasm. This antigen will undergo metabolic events and that processes, molecular or metabolic events, will lead to expression of protein outside the cell, and that is always seen in association with MSc II molecule. MSc II molecule is a part of ACP itself. So in that, here I can say that MSc II and the processed antigen, it forms a complex. So now this complex will be interacting with immature T cells, right? So the entire complex, that is ACP, containing MHC II molecule holding the processed antigen in association with immature T cell will form a complex, and this complex will lead to show certain changes into helper T cell or the immature T cell. That is, this T cell, it will get activated and it will enter into the phase of maturation. Once it enters into the phase of maturation, there is release of biological secretions in the form of interleukins, which are namely defined as interleukin 2, 4, 5 and 6, right? Now these interleukins, I have not mentioned about interleukin 1 to 6. So this interleukin 1 to 6, they are secreted only with the help of activated T cells, that is CD4 cells or helper cells. Now these interleukins, they bring about co-stimulation, and this co-stimulation will lead to proliferation of T cells, as a result of which large number of T mature cells will be generated, and these T mature cells will interact with interleukin 2 receptors, and that will help in stimulating the B cells. So these B cells, when it interact, this is the B cell which is interacting with the T helper cell, will get converted into memory cell or the plasma cell. So here you can see I have shown the formation of plasma cell. These plasma cells, they will start synthesizing large amount of antibodies, right? Whereas the memory cell, it goes into the secretion. So this is how the antibodies are synthesized. So before closing down of this lecture, let me brief you again the process of antibody synthesis. Here what you need to understand is there is presence of antigen which is stimulating the lymphocyte, that is T lymphocyte, and this T lymphocyte will trigger a response to B lymphocyte. But T lymphocyte by itself, it cannot show any interaction unless and until it comes across the activated immunocompetent cells, which are called as APC, that is antigen presenting cell. And these antigen presenting cells are unique because their MHC II molecule, it contains the processed antigen. So how this processed antigen is observed in association with MHC II? Because the antigen is been engulfed by the macrophages or the dendritic cell, wherein the metabolic activities of the antigen will lead to expression of the antigenic proteins, which will always be seen in association with MHC II molecule. Therefore, such activated antigen presenting cell will cause the maturation of T cells, which leads to formation of mature B cell, and these mature B cell transform themselves by the action of clonal proliferation and you can say blast transformation, leading to synthesis of plasma cells and memory cells. Memory cells goes into the secretion, into the circulation, whereas plasma cells goes into the secretion, leading to formation of large quantity of specific antibodies, right? Thank you so much.